Hello friends, welcome again to our channel. In today's video we are going to study about the topic pharmaceutical impurities. So let's start. Pharmaceutical impurity. In pharmaceutical analysis, impurity refers to any substance that is present in a pharmaceutical product which is not the desired active pharmaceutical ingredient and may have the potential to affect the quality, safety, or efficacy of the product. Impurities can arise from various sources, including the manufacturing process, raw materials, degradation of the IP, and interactions with packaging materials. Next is types of impurity. Impurities in pharmaceuticals are categorized into different types based on their origin and potential impact. First is organic impurities. These impurities are typically related to the chemical structure of the active pharmaceutical ingredient and can arise during the synthesis or manufacturing process. They include starting materials, intermediates, reaction byproducts, and degradation products. Organic impurities can potentially affect the purity, stability, and safety of the final product. Second, inorganic impurities. These are impurities that do not contain carbon and are often introduced through raw materials, solvents, or manufacturing equipment. Examples include heavy metals, residual catalysts, and inorganic salts. Inorganic impurities can pose serious health risks if present above acceptable limits. Third, residual solvents. These are volatile organic compounds that are used during the manufacturing process and can remain in the final product. They are regulated by guidelines to ensure their levels are safe for consumption. Fourth, microbial impurities. Microorganisms like bacteria, fungi, and viruses can contaminate pharmaceutical products during manufacturing, packaging, or storage. These impurities can compromise the product's safety and sterility. Fifth, genotoxic impurities. These are impurities that have the potential to cause damage to genetic material, DNA, and can lead to mutations or other adverse effects. Strict limits are set for genotoxic impurities due to their potential long-term health risks. Sixth, degradation impurities. These impurities are formed when the API undergoes degradation due to factors like heat, light, humidity, or interactions with other substances. Degradation impurities can impact the product's stability and shelf life. Note: pharmaceutical regulations, such as those outlined by agencies like the U.S. Food and Drug Administration, FDA, and the International Council for Harmonization of Technical Requirements for Pharmaceuticals for Human Use, ICH, provide guidelines and limits for acceptable levels of impurities in pharmaceutical products. Manufacturers are required to identify, quantify, and control these impurities to ensure the safety, quality, and efficacy of the final pharmaceutical product. Now, sources of impurity. Impurities in pharmaceuticals can originate from various sources throughout the drug development and manufacturing process. Identifying and controlling these sources is essential to ensure the quality, safety, and efficacy of pharmaceutical products. Here are some common sources of impurities. A. Raw materials. The quality of raw materials, including starting materials, reagents, solvents, and excipients, can significantly impact the presence of impurities. Contaminated or substandard raw materials can introduce impurities into the manufacturing process. B. Synthesis and manufacturing process. The chemical synthesis of the active pharmaceutical ingredient, API, involves various reaction steps, intermediates, and catalysts. Each of these stages can potentially introduce impurities if not properly controlled. Reaction byproducts, unreacted starting materials, and other chemical entities formed during synthesis can contribute to impurity levels. C. Reaction byproducts. Some reactions involve multiple steps or intermediates, which can produce byproducts that are different from the desired API. These byproducts can be impurities if they remain in the final product. D. Degradation. Over time, pharmaceutical compounds can degrade due to factors such as heat, light, moisture, or interactions with other chemicals. Degradation products are often impurities that need to be monitored and controlled. A. Packaging in container materials. Impurities can leach into pharmaceutical products from packaging materials, such as plastic containers or glass vials. Substances from the packaging can interact with the product, leading to contamination. F. Cross-contamination. During manufacturing, there's a risk of cross-contamination if equipment is not properly cleaned between batches. Residual materials from previous batches can become impurities in subsequent batches. G. Microbial contamination. Microorganisms like bacteria, fungi, and viruses can contaminate pharmaceutical products during manufacturing, handling, or storage. These can be a significant source of impurities, especially in products intended for injection or internal use. H. Residual solvents. Organic solvents used during the manufacturing process may not be completely removed, leading to their presence as impurities in the final product. I. Interactions with other drugs or excipients. In some cases, interactions between the API and other components of the formulation, such as excipients or co-administered drugs, can lead to the formation of impurities. J. Environmental contaminants. Environmental pollutants or contaminants can find their way into the manufacturing process, especially for natural products or products derived from biological sources. K. Transport and storage. Improper storage conditions during transportation or warehousing can lead to degradation and the formation of impurities. Next, impurities due to instability of the product. A. Certainly, instability of a pharmaceutical product can also contribute to the formation of impurities. Instability refers to the tendency of a drug substance or product to undergo chemical or physical changes over time, which can lead to the generation of impurities. These changes can result from various factors, including B. Temperature and humidity. Elevated temperatures and high humidity levels can accelerate chemical reactions, leading to the breakdown of the active pharmaceutical ingredient, API, and the formation of degradation products that act as impurities. C. Oxidation. Exposure to oxygen in the air can cause oxidation of sensitive drug molecules, resulting in the formation of impurities. Oxidative degradation can lead to changes in color, odor, and efficacy of the product. D. Photodegradation. Pharmaceutical compounds can degrade when exposed to light, especially ultraviolet (UV) light. Photodegradation can result in the formation of impurities that can impact the product's stability and quality. A. pH changes. Changes in pH can catalyze chemical reactions that led to the degradation of the API, producing impurities in the process. F. Hydrolysis. Hydrolysis involves the reaction of a compound with water, often resulting in the breakdown of the molecule into smaller fragments and the formation of impurities. G. Polymerization. Some compounds can polymerize under certain conditions, leading to the formation of larger molecules or aggregates that act as impurities. H. Cyclization. Intramolecular reactions can lead to the formation of cyclic compounds, which can be impurities if they are not part of the desired chemical structure. I. Isomerization. Isomerization involves the conversion of one isomeric form into another. Isomeric impurities can form due to such conversions, affecting the product's purity. J. Hygroscopicity. Some compounds are hygroscopic and readily absorb moisture from the environment. Moisture-induced degradation can result in the formation of impurities. K. Metal-catalyzed reactions. Trace amounts of metal ions present in the formulation or from packaging materials can catalyze chemical reactions that led to degradation and impurity formation. L. Interaction with excipients. Excipients used in the formulation can interact with the API, leading to chemical changes and the generation of impurities. M. Interactions with container materials. The interaction between the product and the container, example, leaching of container materials into the product, can result in impurity formation. Note: stability studies are conducted during the development and post-approval phases of a pharmaceutical product's life cycle to evaluate the effects of various factors on product stability. These studies help identify potential degradation pathways and impurities that can form over time, enabling manufacturers to design appropriate formulation strategies, storage conditions, and packaging to minimize instability-related impurities. Last is control of impurities. Controlling impurities is a critical aspect of pharmaceutical manufacturing to ensure the quality, safety, and efficacy of the final product. Effective control measures are implemented at various stages of drug development, manufacturing, and quality assurance. Here are some strategies for controlling impurities in pharmaceutical products. First, raw material selection and testing. Under this, ensure the quality of raw materials by sourcing from reputable suppliers and conducting thorough testing for impurities. Establish specifications for raw materials that include acceptable limits for impurities. Second, process optimization. Under this, develop and optimize manufacturing processes to minimize the formation of impurities. Use appropriate reaction conditions, catalysts, and purification techniques to reduce impurity levels. Third, synthesis and manufacturing controls. Under this, implement good manufacturing practices, GMP, to prevent cross-contamination and ensure proper cleaning of equipment between batches. Monitor critical process parameters to prevent the formation of impurities during synthesis. Fourth, quality control and analytical techniques. Under this, develop robust analytical methods to detect and quantify impurities at trace levels. Use advanced techniques such as high-performance liquid chromatography, HPLC, gas chromatography, GC, mass spectrometry, MS, and nuclear magnetic resonance, NMR, to identify and quantify impurities. Fifth, impurity profiling and risk assessment. Under this, conduct comprehensive impurity profiling to identify potential impurities and degradation pathways. Perform risk assessments to prioritize impurities based on their potential impact on product quality, safety, and efficacy. Sixth, stability studies. Under this, conduct stability studies under different conditions, example, temperature, humidity, light, to assess the formation of impurities over time. Use stability data to establish shelf life and storage conditions for the product. Seventh, formulation strategies. Formulate products to enhance stability and minimize degradation pathways. Use stabilizing excipients and antioxidants to mitigate the formation of impurities. 8. Packaging and container selection. Under this, choose packaging materials that are compatible with the product and do not introduce impurities. Conduct extractable and leachable studies to assess potential interactions between the product and packaging materials. 9th. Cleaning validation. Under this, establish cleaning procedures and validate them to ensure that equipment used in manufacturing is free from residues of previous batches. 10th. Regulatory compliance. Under this, follow regulatory guidelines provided by agencies like the FDA and IC to set limits for specific impurities. Ensure compliance with relevant pharmacopoeial monographs and compendia. 11th and the last is quality assurance and documentation. Under this, implement a robust quality assurance system to monitor and verify the effectiveness of impurity control measures. Maintain detailed records of impurity testing, process parameters, and manufacturing steps. Thanks for watching this video. If this video was helpful, then please kindly subscribe our channel to get the notes-like content. Thank you.